Starting camera in 3. Oh, no, I started now. Syncing up. 3, 2, 1. Okay. All right. Let's get this party started. Organelles and enzymes. Finding my camera, finding you. And let's jump right into it. So, first off, we've got to hit us with an old recall. Recall. Good stuff. Recall. So, internal cell anatomy. We looked at this in no small amount of detail. We've started to break down the individual components of the cell. What we're essentially trying to do is we're trying to understand both what the biological functions of these different parts of the cell actually are and how they contribute to keeping us alive. And also we're trying to understand what the actual chemical mechanisms that perform these functions are as well, right? It's all about discovery. It's all about trying to understand these different basic mechanisms that are so important to us, to how we function, to how we can continue to exist, and to how I'm able to keep talking and telling you this stuff right now, which is really cool. So enzymes play an integral role in biological organisms. They play an integral role in life, period. Enzymes, of course, technically speaking, they perform catalysis. This is just the name for accelerating a chemical reaction. They accelerate chemical reactions. But because chemical reactions are really just ways of combining sort of the basic building blocks of almost all of our matter, what we're really dealing with most of the time is, in fact, not just catalysis, but something actually a lot more interesting, a lot more specific. We're dealing with functionally the production, the creation of different parts of life. And those different parts of life that we're usually concerned with in biology and in chemistry are proteins. Proteins are chemical molecules that are fundamental parts of how you exist. And one of the things that we're going to explore in this cell chemistry unit is exactly how that functions and how that matters. So, enzymes are super cool. Enzymes perform catalysis. Enzymes are genuinely, for humans, they're kind of like magic. If we extract them from all sorts of different plants and animals, we can use them to do things that we can't figure out how to do in a lab any other way. It's that simple. So, enzymes are super cool. Organelles are also super cool. Organelles, if you remember, they're sort of our basic, they're like our organs of the cell. Organelles are also specialized for particular tasks. Some organelles have more general tasks. Some organelles have more specific tasks. And the extent to which any cellular or biological mechanism is truly specific is a really fascinating question, and something that is an ongoing field of research in advanced chemistry and advanced biology. So, specialized for particular tasks. So are enzymes. And so you might anticipate a link between enzymes and organelles, and of course, you'd be totally correct. Some enzymes are therefore even specialized to particular organelles. And of those few, there's even some that can give us a clear indicator that there is the presence of a particular organelle in a sample. So what we can do is we can chemically analyze essentially water, and we can figure out what chemical components are there, and that will give us an idea of what sorts of cell are there and what those cells have inside of them. So if we're finding all of a particular sort of marker enzyme, that might indicate to us that we're dealing with a whole bunch of cell matter that's is specialized in terms of energy generation. So it's got a bunch of mitochondria there. Or by the exact same sort of limitation, if we're dealing with some particular chunk of cell that has a whole bunch of lysosomes in it, maybe that part of the cell is breaking down some section of the cell, because we can spot those particular marker enzymes for that particular organelle. That's the basics. So some enzymes are specialized to particular organelles. Some of these are marker enzymes. Marker enzymes. Very basic. So what we have going on here with our marker enzymes is just a way of identifying sort of roughly what this section of cellular fluid might actually contain. We can do a chemical analysis on there, and we can learn about some of its biological constituents, because we know that if we find this particular set of chemicals there, that means that we have a particular set of biological structures there as well. Yeah, that's how it works. So, definition. Marker enzymes indicate the presence of a particular organelle. And I will, of course, give you examples of how this works. Indicate the presence of a particular organelle. All right, flip. How am I going to start this guy off? For example, succinate dehydrogenase. So this A's ending, wibble wobble, this A's ending at the end here, very important. Very important. This A's ending, very important. Perfect. Yeah, much better. This A's ending, very important. This, of course, indicates to us that this is an enzyme. So I haven't made any huge mistakes. I'm not talking about a non-enzyme. Succinate is the molecule that this enzyme works on. So succinate is some molecule that we're going to look at in a second. And then we've got some sort of complex here, which is the enzyme. And this complex performs how we name the enzyme. So we named the enzyme dehydrogenase. So that means it performs a hydrogen. It removes a hydrogen, essentially. Dehydrogenase, remove a hydrogen. Hey. Now, there are, unfortunately, lots of different hydrogens in lots of different places, all around all the different things. And as a result of this, fantastic. As a result of this, each of these, both parts of these names are important. So there's a whole bunch of different dehydrogenases. This one is specialized for succinate. And that matters because functionally how this is working is, again, we should think of a physical interaction between a shaped chemical and its receptor here, and the receptor takes on that chemical, and then a crazy transformation occurs inside of the enzyme, and that enzyme's physical transformation tears off a hydrogen somehow. Right? That's our basic concept of how enzymes work. Removes a hydrogen. So, specifically speaking, the chemical, we can model this process with a chemical reaction. Keep in mind this means reaction. We've talked about this before. We can model this process using a chemical reaction, and to do so, obviously, we need to set up our chemical constituents. We have, obviously, an acetic acid here. This is actually kind of a funny guy. COO minus. Very simple. We use the name succinate for this. It is also called, let me look this up, butane-dioic acid, or 1,4-butane-dioic acid, 1 and 4 being the names of the carbon backbone. So the carbon backbone in this case can be labeled either from the top or the bottom because this molecule is symmetrical. 1, 2, 3, 4, or 1, 2, 3, 4. So 1, 4 di is 2. Butane. I don't want to do that bit. But the dioic section tells us the position. The 1,4-dioic section tells us the position of these carboxylic groups. So it tells us that on the 1 and 4 carbons, we have these two OO minuses. Now, this guy goes through in chemical reactions when we're involved with enzymes. What we do is we just draw a big arrow, and then the enzymes act as a catalyst, and where you write catalysts in chemistry is right here. So this goes for enzymes, and it also goes for any other sort of catalyst that you happen to be using. We write our succinate dehydrogenase. Pretty cool stuff. There's also There's another guy who leaps in here. This is part of something. This is part of the Krebs cycle, citric acid cycle, I believe, which is part of the essential set of chemical processes that generate that are involved in cellular respiration, that generate power inside of your cells, basically. Look, there we go. Okay. The product of this reaction is, shockingly enough, going to have less hydrogen. Start with our COO minus. Whoa, did I draw a double bond there? A crime! I was confused there for a moment. I was like, whoa. Excellent. Forgive my double bond, I've sinned. So, here the overall transformation is apparent. We've removed not just a hydrogen, but 2 hydrogens, which is pretty cool. And those hydrogens have been removed from these particular 2 and 3 carbons. So 1 hydrogen coming off this side when it goes through the enzyme, 1 hydrogen coming off this side. The product of this has to be a double bond between these 2 carbons. Yay! So this is the function of dehydrogenase. This particular step occurs as one part of a very big, complicated puzzle that somehow generates a vast quantity of ATP for your body and allows you to continue to live. We're only focused on one tiny little section of the cellular machinery, and we're going to take a little closer look at what actually succinate dehydrogenase and take a closer look at sort of how it physically appears and how it could function inside of your body, because that's what we're trying to do here, man. That's a good time. The product for this is called a fumarate. Fumarate. So that means that overall this reaction takes a succinate, which is also called 1,4 butane dioic. Did I miss something? Nope, just spelling poorly. Dioic acid. Sorry, your standard sort of spelling brain things don't work very well on these words because they're kind of clunked together. 1,4-butane dioic acid. 1,4-butane dioic acid. The butane section. We'll break this down very simply for you. The butane section just tells us this is a 1, 2, 3, 4 carbon backbone. Double check the note. We talked about this before, and then I've already ran over dioic acid for you. You should be able to fully understand where we're going from here. Gosh, take a look. Double check. Gosh. Okay. Now that we're done all the silly mouth noises, it turns it into a fumarate. Fumarate, of course, also has a funky name. Fumarate is also known as, 2E but 2 N-dioic acid. So this is a similar creature, but this 2E tells us that there is a problem, and that problem is something that plagues chemical molecules. We thought when we were labeling them in basic ways in our first sort of little introductory chemistry section that we would have a pretty decent handle even on just simple basic molecules with only like, a short number of carbon backbones. We were wrong. It turns out that there's all sets of what are called isomers. Of all sorts of isomers are essentially just different forms of what appear to be the same molecule when we're dealing with just their chemical formula. This is maddening. So the most irritating form is probably something called an optical isomer or an enantiomer. Optical isomers are visually different because of left and right-handedness, which is maddening to explain. You do some research, find it out yourself. This is a much simpler form of isomer, which we're talking about right now. Unfortunately, this is a type of isomer where it has the same chemical formula, but its physical shape in space is just different. Its physical shape is just different. Physical shape is different. In order to specify which of the different physical shapes it could be, we use this 2E notation. Yeah. Apart from that, you should be able to spot some of the basic parts of the name set up here. Butene is nice and simple here. This tells us that we have on the second carbon a double bond, right? So butene tells us we have a double bond on the second carbon. I've explained the 2E for you as much as I can right now. Hopefully the rest of that name makes sense to you. The technical name for these isomers where the physical shape is different is stereoisomer. I think of the basic concept, the idea that these things with the same chemical formula can occupy space in different ways. Don't worry so much about these specific names like stereoisomer, optical isomer, or enantiomer, enantium. Like, don't worry about that so much. It's much better, much more important that you understand the general concepts, and that when you are reintroduced to the names, you're able to pick them up that much more easily when you need them in that particular context. All right, zoom this page away, and we're going to keep on zooming. So let's take a quick look at what our complex might look like. So succinate dehydrogenase is a part of complex 2. Complex 2 is again part of Krebs cycle, which is this big energy generating cycle in your body. Complex 2 normally, so here of course we have our absolute classic phospholipid membrane. This genuinely is just a perfectly standard start to the vast majority of these sorts of cellular diagrams. Once you spotted it, you're going to see it everywhere. The complex is itself split into these subsections, which I'm not going to get too deeply into. We're just going to draw a rough image of its overall shape, and then I'm going to try and draw a more specific image that looks a little more exactly at the curving of the chemical structure. These sorts of lines and blocks actually give you more information about chemical structure than you might first expect, but it can be kind of hard to see how that would be the case when you're first introduced to it. So here we have the same sort of basic diagram as what I had with my succinate dehydrogenase here. This, I'm drawing the actual complexes, the chemical structures involved in doing this operation. But of course, we still need our succinate coming in and our fumarate coming out. Suddenly doubted my ability to spell that. Succinate coming in, fumarate coming out. The fad H, not the NAD H cycle. The fad to fad H cycle occurs actually inside. Bunk. And you can see this H2 actually balances out the two H's that we're losing in this particular case, right? Both of these H's disappear. They become part of this H2 here. Super cool. So what's cool is this particular thing, which is kind of crazy, operates inside this part of the cell membrane here. Interestingly enough, this isn't the cell membrane that's extra, this most of the time isn't going to be the cell membrane that's extracellular, but it's actually going to be the mitochondrial cell membrane. And this is nifty because it reminds us that the mitochondria itself has its own cell membrane for historical reasons, which is a hilarious thing to say in a biology course. What we have here is a number of parts. How am I gonna really explain this? I think the simplest way to do it is to look at the basic the sort of more complicated diagram and then compare the two. So let me draw most molecules come out either sort of curvy or in plates. And that seems like a radical oversimplification. And it is, because the thing that makes molecules complicated and weird is the fact that they spend an awful lot of time moving around. And we've talked about that a lot in terms of enzyme interaction, but you really can't view our physical reality at the cellular level as anything other than this constant sort of exchange of moving parts and pieces, right? We're not static. We're incredibly fluid and moving, and that's really, really cool, and it's an integral part of how we function as people. I'm trying to draw these units roughly accurately. You'll notice it's all sort of swirly and crazy. There's more. It should hopefully be apparent that there's more like space available in this particular one. That means that there's actual space available for it to grab onto these necessary molecules that it has to engage with, which is really cool. It has to be able to physically interact with the succinate. It has to be able to physically interact with the fat and the fat H2 in order to carry out this operation. So this is sort of how it actually looks. And now let me try and sink it down into the cellular membrane for you. I'm going to draw the tails of all these sort of overlapping forest of lipid ends, because remember, our lipid ends have sort of different shapes and different structures, and if we're looking at different layers of them overlapping as opposed to just a single layer here, then we definitely get this like thick, dense forested effect, which is cool. They also have really, of course, important chemical properties that are making a radical difference in enabling much of this process to occur. Part of the reason that this goes on inside of the membrane is because it creates this optimal set of conditions within which one of the products of this process can continue on into the Krebs cycle, can continue deeper on into the processes that drive our body. So this is a pretty good example. Here we have the physicality of the molecule laid out. The one major error I think I've made here is that ideally, let me use a pencil. Ideally, this section would be a different color to sort of indicate the fact that we're dealing with a different subunit here. These overall sections are then obviously much easier for us to deal with. You can see why there's a preference for this sort of diagram over this sort of diagram. This sort of diagram may be more physically accurate, but because of the complexity of chemical molecules and the fact that they overlap, it tends to obscure as opposed to actually make things simpler for us. So this is one really basic, good example of how organelles can have specific marker enzymes associated with it. Succinate dehydrogenase is the marker enzyme for the mitochondria, and the reason for that is because it plays this essential role in an essential process that the mitochondria performs. So the mitochondria needs a lot of them. Succinate dehydrogenase is the marker enzyme for the mitochondria because of the essential role it plays in mitochondrial function, and its ubiquity, which just means the fact that it's everywhere. And it is very common in the mitochondria. That's it. That's all you really need in order to be a marker enzyme. So that's a very satisfying situation to be in. We have started to weave together these different individual sections of biology and chemistry. Now we're going to take it one step further, and we're going to look at free-floating enzymes. So here we have an enzyme that is locked into the cell membrane, and this is how a lot of enzymes function. But a lot of enzymes are also released into particular three-dimensional areas inside of the cell, whereupon they wreak havoc. Some of them quite literally. We've been thinking of enzymes as very constructive, but they also are play huge roles in decomposition. They break things apart, right? Also very important. Synthesis and decomposition. Those are sorts of the two types of reaction that I failed to remember at one point, but of course, now that I'm not talking about them, they're on the top of my head. That's just how things work. So let's do some forensic biochemistry. Forensic biochemistry. Ooh. Enzymes can be rooted or they can be free moving. Or free moving. Usually they're going to be rooted in some sort of cellular membrane, but they can also be rooted in a number of other things. The ways in which enzymes are constructed into complexes is complicated, incredibly complicated, because we're dealing with overlapping cellular pathways, which is always a huge issue. So, for example, enzymes can be rooted or free moving. For example, purple acid phosphatase. Phosphatase. Phosphatase, nice and clear what that is supposed to do again. Right? Phosphatase removes phosphate. That's going to be the guess. Or it adds phosphate. Sorry, there's no D on there. Whoa, slow down, slow down. Right? Enzymes are nice because there's so many of them that we tried to name them in relatively achievable ways. Well, let's find out if this is. Let's find out if this is true. It either adds or removes them. We're gonna do it ourselves. So this is the chemical reaction that Pat performs. Reaction. Purple acid phosphatase. It takes something called a P ester, which looks like this. This R stands for the rest of the molecule. This is because P esters are actually a class of molecule. They're not just one specific molecule. Important to understand that. Here we have our oxygen and hydrogen, of course. This is our phosphorus at the center, gotta be. And it's attached to another oxygen and a hydrogen. These oxygens are really important. They play a prominent role. Yeah. Oxygen is, it turns out, really a huge deal. Oxygen, carbon, hydrogen, we're starting to see the repetition of these very basic subunits over and over again. That's no coincidence. Plus H2O, there's two components here, so we got to get some water in here as well. That's almost never going to be an issue for your cells. Your cells are chock full of water, or you're dead. It's basically that simple. Here's our PAP, or purple acid phosphatase, doing its work. The product is some sort of alcohol. Alcohol is just anything with an OH on the end of it. Human drinking alcohol is only one particular type of alcohol. It's an ethanol. We can also drink methanol, but it kills us, so don't do that. And way faster than regular alcohol. I know regular alcohol kills us too, but to a lesser degree. Methanol bad, ethanol good is the rough thing. Well, good is a strong term, but certainly in terms of speed of death. That should not be how you measure quality of things as a general rule. How fast it kills you should not be how good or not good the thing is. That's my general rule. Cool. So, this is kind of a cool piece. It's called phosphatase because of how it acts on phosphate. I would probably be happy calling it like dephosphatase or something. I'm not sure why they did that. It kind of does remove a phosphate, but I think it's better to call it splitting. Here it splits. So it splits off this R group, which is the rest, from, in terms as an alcohol, it splits off this R group, the rest, from this simple phosphate. Right? So that's a nice setup. Here we have something that you can kind of start to see how it might function. And what we're going to do, the reason this is forensic biochemistry, is we're going to try and put this purple acid phosphatase back into its context in order to try and understand what role it's actually playing inside of the cell. Let's get on our second page here, doobly-doo. So here's our basic setup. We have a plant cell, as you can tell, because it's got this big, thick cell wall. Yeah, that's good stuff. Inside of it, you have a number of processes, including, it's got a big vacuole in it. Vacuoles are essential for keeping plant cells rigid. If you don't feed everybody knows that if you don't feed your plants water long enough, they start to go limp. The reason for that is because they have these large vacuoles inside of them that are giving each of their plant cells tension. And those vacuoles are filled with water. When the water is full, then the plant is nice and rigid and tense. When the water starts to reduce, the plant physically gets weaker and less filled with water, and therefore, like a balloon, it starts to deflate. Makes a lot of sense. Which parts of these are you going to draw? Okay, so this is emitting something over here. And it's emitting PAP over here. And here's where our flow of P esters is coming from. So there's a number of different things occurring at this cell all at the same time. One of the things that is interesting is I believe the PAP is actually being created inside of the central vacuole, which is super cool. It gives us an indicator that this vacuole is functional, right? It's not just empty space inside of the cell. There's actual useful things occurring inside of there as well. So PAP is being manufactured inside of the vacuole and then released into the wild. What's interesting is then it's acting on these P esters and then converting them. It's cutting off these long chunks of them, and it's producing lots of just random phosphate groups, right, which we're going to call PI. So all of these random phosphate groups are kicked out of this particular weird, crazy reaction thing that's going on. And then also over here, we have the function of another enzyme. This enzyme is phospholipase. Phospholipase. This is cool. This is obviously another enzyme. We're starting to spot the enzyme. But what this enzyme does is it's part of an entire class of enzymes. Is another class of enzymes that can break down phospholipids. So what we have here, if I'm understanding this article correctly, which I hope I am, what we have here is this really cool combination between these two enzymes. We have the phospholipase going out and breaking down what appears to just be phospholipids, parts of the cell membrane, and then as a response to that, those are sort of like dirty phospholipids though, because they still have combinations of phosphorus and then a lipid ending. So then you also need a piece that comes along and like gets rid of just the phosphate, right? So that's the role that our PAP is playing. Our purple acid phosphatase. Purple acid phosphatase. Right? That's the role that our PAP is playing. Our phospholipase is the one going out there, and it's like the big sword that slices, just like cuts things out. And then there needs to be like a smaller weapon to go through and like break up the individual little pieces. The cell in this particular case is acting kind of like a butcher. It's got a big knife doing big chops, and then it's got a little knife doing little chops. Super cool. So that's a bit of sort of cellular forensics for us. Just by sort of looking at a couple basic enzymes that a cell is emitting, we can start to get an idea of what role this cell might be involved in. Clearly, there's some sort of recycling effort being involved here with the phospholipase. One really interesting question is this, where are these cell walls coming from? Cell membranes coming from? Where is this section of cell membrane coming from? Is this an external part of the cell that it's breaking down, or is it an internal part of the cell, a cellular mechanism? Is it taking something from inside the organism and recycling it, or is it taking taking something from outside the organism and digesting it. That's where we need to understand the context of a particular part of cellular machinery in terms of the biological organism as a whole. And that's why biochemistry is such a natural combination at a certain point, right? If you want to understand the particular type of organism and a particular type of function to a particular level of detail, at a certain point, if you can, you're going to want to break down into the actual chemistry of the thing. Let me see if there's anything else cool I want to say about phospholipase. Yeah. So the last thing I want to say is all I've said here is just a suggestion, right? So I've suggested recycling rolls for PAP and phospholipase. In order to confirm that, but I don't know, right? And that's okay. I don't have to know everything, man. The reason we're bringing this up, though, is in order to confirm that, we need to use science. And specifically, we need to use the scientific method. And that means that we need to actually go out and perform an experiment. We need to actually go out and do specific, rigorous research on this question, or we need to find somebody else who's done it for us. Do rigorous experiment, or find someone who has. Right. So all I've done here is I've given you a diagram of a cell, and we've talked about a couple different things that I've got it under good authority that this cell emits, and then we've tried to fit that into our overall understanding of cell chemistry. Because you're already familiar with cell membranes and phospholipids, your ability to do this is pretty much the same as mine. But you have to remember that we must be very conservative when we're making scientific claims. The only time we can make a really accurate scientific claim is when we have scientific evidence or proof. And that is found by doing rigorous experiment or by finding evidence of rigorous experiments that have been done. That's it. That's the essence of science. That's the essence of chemistry. Hope you're enjoying these sets of lectures. Next up, we're going to tackle one of the most important topics in all of biochemistry, the genetic code. Have a great day. Thank you so much for watching.